So back to our channel. Today we are going to start our embryology. So we'll complete embryology playlist. Try to complete all the lessons. So first let us check out some preliminary considerations of embryology. So we know that embryology is nothing but the study of development of an individual before birth, that is prenatal life, not after birth. So this whole book is only about prenatal life, that what happens. So the first 2 months when the baby is developing, it is known as embryo, and after the 2 months only, that is from the third month starting, we call it as fetus. So remember, before 2 months it is embryo, and after 3 months it is fetus. And these common things, we know that testis is a male sex organ and male gonad, and the ovary is a female sex organ or female gonad. So they mainly produce gametes. So the male gametes are produced by testis called as spermatozoa, and this process is also known as spermatogenesis. And female gametes are produced by ovary called ova, and the process is oogenesis, and spermatogenesis and oogenesis together called gametogenesis. So these are like highlights of this lesson. So first, if we see the highlights, we can discuss in detail of cell cycle, all those introduction. So after completing highlights, we'll see those details, and after details we'll see all the diagrams in our first lesson, so that we can complete it perfectly. So fertilization is nothing but, we know, when a ovum fuses with the sperm and forming a zygote. So the order follows like first the zygote forms, next the embryo, that is up to 2 months, and from the third month the fetus. So zygote, embryo, fetus. So that is the order. And the characters of the parents are transmitted to the offspring. Yeah, we know by DNA, that is genes are such strands of DNA. Genes are made on such strands, so they are located on the chromosomes. So remember, this is important, that a typical cell mainly contains 46 chromosomes, that everyone know, diploid number, that is 2n condition, but a gamete contains only 23 chromosomes. Zygote forms, that is male and female gametes. So again that diploid number is restored in the case of fertilization, right? So multiplication of cells just takes place in normal body cells, that is mitosis, but we know meiosis takes place in a germ cells to form gametes, right? So special kind of cell division takes place, that is to form gametes, is our meiosis. So remember, in mitosis only 2 daughter cells will form, and in meiosis 4 daughter cells will form. And remember that every human is from mother, who in mother we have stayed for first 9 months, that is equal to 260 days, 266 days, or 38 weeks to be exact. So 38 weeks, 266 days, we are in our womb. So just these are like important. So now let's begin with our cell cycle first. So why we are starting with cell cycle? Because we know this embryology is all made up of like from single cell zygote, how a human body is going to produce. So we need to know some details of cell cycle to understand this lesson. So cell cycle, first one, let us take mitosis process. We have mitosis and meiosis in cell cycle. So cell cycle is nothing but, first the G1 phase, S phase and G2 phase. So here the G1 and G2 are like preparative phase, and S is a main synthesis or replicative phase in case of mitosis. So in interphase, so cell cycle is divided into mitosis and interphase. So interphase contains G1, S and G2, but the main cycle is the mitosis cycle. So here you can check out like first the cell duplicates the DNA before there is increase. Cell duplicates its DNA in the synthesis phase. So before going this process, the cell need to increase its cytoplasm, produce many proteins, so that it should be ready, so that it can undergo in the synthesis phase. So what happens in synthesis phase, that is this 2n, it converts into 4n, that is double structured chromosomes are formed. And here you can see how the cycle starts. So this is first the G1, that is a pre-synthetic or G1 or gap phase, and this is the S phase, that is the synthesis phase, where we have said here the DNA gets replicated and the chromosomes gets duplicated. So this is important. And after the S phase, we know it goes directly into mitotic phase, and after mitotic phase, finally 2 daughter cells are formed, and 2 daughter cells are formed. So one of these daughter cells may go into G0, that is exit the cycle and stay rest there. Otherwise, whenever stimulated, they can again come back to the cycle. And some cycles are like this daughter cell again continues to continue the cell cycle. So those are like having continuous cycle, but some may also go to rest. So here G2, you can see this is a post-synthetic or pre-mitotic phase, because post-synthetic after synthesis and before mitosis phase. So it is post-synthetic or pre-mitotic phase, and this G2 is also a preparation phase. So the cells with the continuous cell cycle can be seen where, that is without going into this G naught or quiescent or exit cycle. They're continuous in case of our GIT, skin, bone marrow. So these are like examples. So here the cells are known as labile cells because they multiply all the time. Next, coming to the cells which stay in mainly G naught, that is the stable cells. So if they stimulate, they can enter the cell cycle, otherwise not, they'll just take rest over there. So examples are here like hepatocytes, kidneys, pancreas. So here the cells are always in resting. When stimulated, then they come to action. So next, coming to permanent cells. So what are permanent cells is that permanently in resting stage, that they don't want to participate in the cell cycle. So we are interested to see what kind of cells are that. That is in G naught they can't enter the G1 cycle. So example here like permanent cells, neurons, skeletal muscle cells, myocardial cells. So I think the number of mutations going on here would be low because they're not participating in cycle only. So I think no mutations and no cancers over this neurons, skeletal muscle, myocardial. Of course there are, but I think very rare cancers. So next, coming to mitosis, the main cycle here which is going on. So how it takes place, let us see. So mitosis itself is nothing but defined as nothing but the division of nuclear material, that is dividing into two daughter nuclei. The nuclei divide here mainly, and then after that cytokinesis takes place, that is the cytoplasm, all the organelles all gets divided, and finally two daughter cells are formed. So that is known as cytokinesis, right? So we know these wall phases, prophase, metaphase, anaphase, telophase. We are learning it from many days, right? So prophase, what happens? First, the condensation of loose and chromosome. First, at G1 phase, what happens? There is condensation of loose chromosome, right? And next, the chromosome is condensed so that we can see them clearly in the case of prophase, and the nuclear membrane is getting dissolved, and the central centrioles are present at the opposite side, and the laminin protein is phosphorylated, and the membrane gets dissolved, nuclear membrane gets dissolved. So this is prophase. Next, coming to metaphase. So metaphase is nothing but we know that chromosomes are aligned in the midline, that is in equatorial line, and the microtubules start forming along opposite ends of the centrioles. So each chromatid, so what is chromatid? This single structure is chromatid in a chromosome, but both together from the chromosome, and the middle part which is joining this chromatids is nothing but known as kinetochore. So next let us see telophase. Telophase, what happens here? The cytokinesis takes place and the cells start dividing, thereby the cytoplasm. Now nuclear membrane again start forming, and again chromatin gets loosed here. Because in prophase the chromatin is getting condensed and we can see it properly. But again telophase is nothing but like before, like G1 phase, again everything, G2 phase, everything is getting back. Like the nuclear membrane is getting back, and also even the chromatin is getting loose. So the cell cycle is well regulated, right? You can see here like how G1, yes, yeah, everything is well regulated. So if there is any dearrangement or any disturbance, damage, then the G1, in the G1 for example, then they are not allowed to S phase, and it is scanned mainly by checkpoint, G1 checkpoint, also known as G1S checkpoint. And next there is also DNA replication, then only DNA replication takes place, and there is also G2 checkpoint to check the number of replication errors. So G1 is mainly like whether to send the cell to synthesis or not. If any repair, then they'll exit here only. In G2 it's like G2 checkpoint is like after replication also, is there any replication error or not, to check that. So that is G2 checkpoint, or also known as G2M checkpoint. So we are having many cells, like we know there are proto-oncogenes and tumor suppressor genes. This I think everyone knows. So in proto-oncogenes there are like cells which are expressed as receptors, like ligands, like signal transducers, which give message to the nucleus, and these cells in turn give effect to activate genes so that the cell proliferation continues. So there are different mechanisms going on over here. So we know this like for example, if we take a cell from the G1 phase, so let us go deeper into this story and understand. So we know from male and female chromosomes, alleles are like similar, homologous with each other, except the X and Y, right? We know that all 23 chromosomes are homologous to each other except the 24th one in case of male or female. So here what happens is they send mitogens or growth factors or mitogens like outer ligands, and receptor is also found by these cells, and inside signal transducers is also found by these cells. So in turn which forms a like cyclin dependent kinase, and cyclin dependent kinase will drive over this process with RB, that is retinoblastoma. Retinoblastoma is one of the tumor suppressor gene. So when this retinoblastoma is activated, it releases the E2F transcription factor, and this E2F leads to proliferation of cell. So usually retinoblastoma is inactive. So remember, when the retinoblastoma gets inactive, then only DNA replication takes place, because it is phosphorylated and become inactive. Then only E2F is released. Then only our uh, proliferation takes place. Why it is inactive? I think you can guess it by now, because retinoblastoma is tumor suppressor. Tumor suppressor means it is decreasing the cell cycle. So if it is inactivated, then only cell can undergo proliferation, right? So when the cancer may develop in this kind of cells is when the proto-oncogenes are converting into oncogenes, that is any mechanism in this gets altered, like leading to more amount of proliferation, then it may lead to cancer. And also the tumor suppressor genes should more suppress, that is they should not work at all, because tumor suppressor genes normally suppress the tumor forming cells, so they should block, then only the tumor can form easily. So remember that the cells which are active all the time are CDK dependent kinase, and they are stimulated so that DNA replication takes place, that is like acceleration. So here you can take like a car example. In car, accelerations are like proto-oncogenes, and if tumor suppressor genes are stimulated, they stop the DNA replication, that is like brakes of the car. So we can analog is like here car. So with that we can explain this. So here let us understand some of examples from here. Like for example, there is G1 at the G1 S, next RB is inactive, then E2F is released, then synthesis takes place. And G factors like platelet, growth factors, different growth factors, example like platelet, epidermal, vascular, epidermal, vascular, endothelial. And receptors are like extra and intracellular domains. Yeah, receptors, we know they're having extracellular and intracellular domains. Examples, we have one pass receptors, etc. In the case of intracellular domain, we have tyrosine kinase, which phosphorylates by autophosphorylation, so that even there are like two receptors, both get attached with each other, and they signal transduce act on the RAS protein mainly. So RAS signals the nucleus by RAF, MAP kinases, etc. So what happens is finally transcription factor is released, so that our replication process continues. And also like one pass receptors, here growth factor act on our receptor, activate Janus kinases, and activate STAT. So STAT is signal transducing and also activating, so that the signal to the nucleus is reached. And also there are 7 pass receptors which act upon G stimulatory, that is cyclic AMP is activated, protein kinase is activated, and again stimulus to nucleus. So these all mechanisms are like examples how a nucleus get stimulated. And another 7 pass receptor may be G inhibitory, which gives response to protein PLC that forms DAG and ionophosphate IP3. So from endoplasmic reticulum, it releases calcium, and calcium kinase is attached to it, and again give signal to the nucleus. So this is like some of the examples how the nucleus gets stimulated. And we are also having here one more like MMC, Gen, REL. These are activated, and these activate the transcription factor. So here receptor, ligand, segment transducers, those are like proto-oncogenes which are stimulated when the cell proliferation occurs. And in tumors, they are mutant, so that cell over proliferate and leading to carcinogenic. So either paternal or maternal allele may be mutant in the case of proliferation, right? At least one allele is mutant, then also it may lead to cancer. It is not a rule like both should be mutant. So RAS activated by GTP mainly, so which gives the signals. So if off, then it will close. It will be attached to GDP, usually RAS. So whenever the RAS gets mutant, that is there is no GDP attached to it, so that it is always on, kept on, and the cell may lead to mutation. So the gain of function of proto-oncogenes can cause cancer. That is, the proto-oncogene converting into an oncogene is very dangerous, which causes cancer for sure. So retinoblastoma is a tumor suppressor protein, so that you need to remember. So in the tumor suppressor protein, we are also having our main that is p43, guardian of genome, and ATP is a scanning gene or DNA, and retinoblastoma, every cell have the retinoblastoma. So what this ATM does is, if any fault in the DNA replication, then the ATM reports to the p53, which activates and dissociates with MDM2. Actually, p53 is always associated with MDM2. So whenever there is any disturbance in a cell replication, then it gets detached from the MDM2 and it goes into action. So what it does? So the p53 activates p21, p57, p27, etc. and destroy the cyclin and CDK complex by the CDK inhibitors. So by this the cell cycle stops and next activator, and it also activates DNA repair gene. So if it is repairable, then it will repair and again start binding with MDM2 and take rest. But if there is so much damage in the DNA, then what p53 do? It allows the cells to go senescence or final order to go pro-apoptotic genes to undergo apoptosis, that is cell suicide. So this is how our mechanism works in the case of any cancer or mutations. So remember that 70% of the cancers are with p53 mutation. p53 is damaged, that is high mutation chances in the case of p53, and this may lead to neoplastic transformation. So this is all about like neoplasm, some of cell cycle regulator process, etc. Now let us go deep into meiosis. So meiosis like gametogenesis, because meiosis is a process which takes in a gamete forming cells. So gametogenesis is divided. We know meiosis is a reduction division. And because why it is reduction division? Because in daughter cells you can see we are having finally only one chromosome, that is single structure chromosome. Actually we start the cycle with double structure, but finally only single structure chromosome. Of course 4 daughter cells are formed, but only single structure. So it is a reductional division with the 4 daughter cells. And remember that the gametogen, and we started with a diploid, but finally they end as haploid. So by this also we can say it is a reductional division. So gametogenesis in male is nothing but spermatogenesis in female oogenesis. So in female the meiosis takes place, cytodifferentiation takes place, finally forming a mature ovum. That is our definitive ovum. And in spermatogenesis, meiosis takes place, cytodifferentiation takes place, that is mature sperm or spermatozoa is finally formed. So let us understand the process meiosis. So in the case of female it is oogonia, in the case of male that is spermatogonia. So whatever the gonia, remember that in male it is spermatogonia B. So first we are having 46 chromosomes coming from our mother and father, right? That is 23 from our mother and 23 from our father. So this is diploid condition, and here the structure is 1n plus n, that is 2n. So one chromatid. Actually this structure is mainly based on chromatid. Here we have one chromatid. So together we have two chromatids, and we are having diploid condition here, that is 46 chromosomes. And remember that 22 chromosomes are homologous with each other, except X and Y, which are not homologous, heterologous with each other. So right now let us start our meiosis. So before undergoing meiosis, actually cell get duplicated, that is chromosome get duplicated, that is one more chromatid adds to it and start the meiosis 1. So what happens in meiosis 1? Let us see, that is primary gamete is formed, that is double structure. The chromosome is duplicated over here. So here the structure becomes 4n now, because according to chromatids, right, capital N, 2 capital N, 2 capital N, together 4 capital N. And here but the condition is still diploid, because the inside DNA content is same. So it is diploid and 4 capital N, and it is nothing much known as primary gamete. So you can see the replicated DNA and the duplicated chromosome is present while starting our meiosis process. So this is a double structure, one chromosome, two chromatids, right? Now next process is synapses. So here what happens, that homologous chromosomes come together. So you can see the two chromosomes are getting together, and here the crossover takes place, which is exchange of genetic material. That is, some paternal part goes to maternal part and some maternal part goes to paternal part, so forming a chiasma over here. In the chiasma only we can find out that in the maternal chromosome we will find some of the paternal chromosome, and in paternal chromosome we will find out some of the maternal chromosome. That is nothing but they both have exchanged their genetic material. So next alignment takes place, that is alignment on our spindles takes place. So after alignment finally they divide, that is forming two daughter cells in the case of meiosis 1. So here the structure is again 2n and only haploid condition. So remember that because they are divided now, it is diploid here, but they have become a haploid here, but double structure. Here also double structure, haploid. So in the case of female, it is known as secondary gamete, that is secondary oocyte with one polar body, but in the case of male, they are forming secondary secondary spermatogonia, right? So one and double stranded 23 chromosome haploid. So here the structures have become haploid. So we are saying it is reductional division. Now the meiosis II takes place further, that is these are again further divided into each chromatid, but remember here one chromatid with paternal, for example with maternal, but it is maybe purely paternal or purely maternal, so like that they are divided into four. So you can see in the case of female it is known as one definitive ovum and two polar bodies are formed, and the first polar body again further divides into 3-4 polar bodies. So all these polar bodies are degenerated by the definitive ovum, finally meets with the sperm and the fertilization takes place. So in the case of male, what happens, all 4 daughter cells are formed and 23 chromosomes, haploid and 1N. So this we need to remember. In the case of male, 4 spermatids are formed and these 4 spermatids will later turn into spermatozoa in the case of male, but in female remember only one definitive ovum and all others are converting into the polar body. So this is all about our mitosis and meiosis general introduction of embryology.